Immune cells, the body's defense against disease. They tirelessly patrol under our skin and throughout our body. They identify and destroy microscopic invaders without us ever knowing. But there is an enemy that immune cells cannot overcome. This enemy infects the immune cells. It decimates their ranks until the body is left defenseless. It is the human immunodeficiency virus, HIV. HIV is the virus responsible for acquired immunodeficiency syndrome, or AIDS. Immunodeficiency means that the body's immune system can no longer protect against infection, including cancer-causing viruses. HIV is contained in the blood, sexual fluids, and breast milk of infected individuals. HIV can spread by direct blood-to-blood contact during blood transfusions or contaminated needle usage. HIV may also infect another individual if any fluids containing HIV come in direct contact with the membranes of the genital or digestive tracts. For instance, within the digestive tract, HIV may cross the membrane through a microscopic injury. Once in the underlying tissue, a patrolling immune cell, known as a dendritic cell, may be the first immune cell to find HIV. Dendritic cells have molecules on their surface to which HIV sticks. The dendritic cells surround and carry the HIV in deep tunnel-like folds in their surface. HIV may infect these patrolling cells or other cells in the tissue environment. The dendritic cell will then find a helper T cell. Normally, the dendritic cell activates the helper T cell, and the T cell emits signals to call in other immune cells to fight an infection. However, when HIV has invaded, the patrolling cell is carrying an insidious enemy, intact HIV virions. The helper T cell is their primary target. The T cell may extend parts of its surface into the dendritic cell's folds. HIV now has access to the T cell membrane. The helper T cell is HIV's ideal target because the membrane has two molecules that it needs to enter the cell, CD4 and CCR5. The T cell normally uses these molecules to interact with other immune cells. On HIV's own membrane is its only surface protein, the envelope glycoprotein spike. The spike has 3 outer subunits that conceal 3 inner subunits. The outer subunits encounter and bind to CD4, tethering the virion to the membrane of the T cell. More spikes begin to attach. As the outer subunits bind to CD4, they rotate outward. CCR5 can now bind. The inner subunits are now revealed. They extend their special attachment portions called fusion peptides. The fusion peptides latch onto the T cell by inserting into the membrane. The inner subunits then fold and twist, pulling the membranes together. This event is called membrane fusion. HIV empties its capsid and proteins into the cell. The capsid contains viral RNA, which is a set of molecular instructions to make more HIV. In a complex multi-step process, HIV's proteins put the viral instructions into the DNA of the T cell. The T cell is now infected. When the cell uses its DNA, it will read the viral instructions hidden there and produce more HIV. If left untreated, HIV will eventually deplete the number of T cells in the body over the course of several years. When the immune system no longer works, the condition is called AIDS. Other diseases can take advantage of the weakening immune system, leading to severe illness. Unless treated, an individual with AIDS will die. There are drugs called Antiretroviral Therapies, or ART, that can slow down HIV infection. However, a pill containing a combination of 3 antiretroviral drugs must be taken every day, and the infection is nearly impossible to cure. Worldwide, there are currently 34 million people living with HIV. Only about 10 million have access to ART. It is estimated that about 2.5 million people are newly infected each year. In 2011, 1.7 million people died from AIDS that developed due to HIV infection. The scale of the HIV pandemic and its link to cancer make it one of today's most difficult health challenges. Researchers are trying many strategies to stop HIV. One strategy is finding molecules that can interfere with the envelope glycoprotein and prevent HIV entry. Once a molecule is discovered, scientists will try to develop a drug. Since 1987, there have been 187 HIV vaccine trials. None have been successful enough yet for a global vaccination strategy. A vaccine is preferred over other prevention methods because it only requires a one-time administration. After vaccination, a person's own immune system can recognize HIV and respond quickly before HIV establishes an infection. Recently, a vaccine specific to one HIV strain was found to be moderately effective. There is hope that this vaccine will lead to the development of vaccines to more strains of HIV. In labs across the world, the search for solutions continues. Understanding the biology of HIV and its envelope glycoprotein will be key to ending the pandemic.